is a national assignment. Okay, it's not an issue of somebody saying this is, I have to do everything by myself. Okay, so there must be that synergy. It's that synergistic effort that our DG is trying to come up with. Thank you.
So let me call on the second presenter of the day.
Okay.
Is it in that operation within the ambit of the Federal Ministry of Health? To what extent are you collaborating with the ministry, especially the Department of Food and Drugs? Because at one stage, NAFDAC was above the ministry. Now, if NAPRID is also strong, they will go above the ministry. And yet you need a focal ministry. You need to operate within that environment so that you get all the support. Otherwise, you'll be on your own. Somebody must be there who can say, NAPRID come, PCN come, this one come. But if you are on your own, you cannot directly go and invite other agencies to come and work with you. It will be more difficult. And that is true. So I want to ask that when we're planning, we should plan holistically. Take everybody along. If you have the resources, that will be good. That will make you even the focal point. Are we together? That will make you even the focal point, but you must carry other people along and, appreciate the hierarchy of the government. We do not operate in vacuum, but that is breaking down very seriously because everyone that gets access to money decides to go in, become independent. And as far as I know, unless there is this synergy, you cannot make it. And that is dragging us back. Please, I appeal to you, tell the DG. Prof, please tell DG that he should work within the group and make sure that, everybody is carried along. Otherwise, we're going to have problems. The money will be wasted. And then in the end, we will not have any results to show for it. It's very important. Everyone who makes his efforts will be accounted. Will be accounted. Somebody will recognize him. It's not about one person taking all the credit. It's about everybody taking the credit. And within that group, we still know who gets the greater glory. So I'm appealing, please. Thank you, sir.
Thank you. I think this one is a home take home a lesson for us. I think okay. Thank you very much, Father Adagazu. I've been asking him what he has been taking. He didn't understand. I told him that he said whether he knows beauty. I said, no, you can't know me. Those days we are very, very small. You understand? Yes. That's okay. Look the difference. Thank you very much, sir. With respect to this, carrying everybody along is very, very important. Because if you also check with what I read, the DG speech, he included it there. There's no how we can succeed with this without NAFDAC, because they are going to prepare the guidelines for safety, for quality, must be there. And for us to bring a vaccine that will be, we will have vaccine party and all those things, they must be there. We also must carry other stakeholders, Ministry of Health, NIMA, and so on and so forth. Everybody must be there. And that is also why we said that during the last engagement, it was said that we have 3 years to do this. We said, good, but there must be a strong committee of M&E that is going to, every step, every year, to tell us how far we've gone. And just like this stakeholder, NAFDAC is here and everybody's here, maybe that one will be more robust. So it's not going to be a one-man thing, because this vaccine thing is not like politics. So look at what happened during COVID. Every man to his tent. We don't know the another disease that is around the corner. Nobody knows. So the more we start this thing and own it, yes, not only the vaccine for MRI, there are emerging technologies that we can be able to go fast to develop vaccine for any emerging disease. And that is what, as a country, we should be running. When you talk about health, you should not be doing politics. When you talk about defense, you should not do politics. So health, we cannot do politics with this. So that is the stuff. And the DG has also said, because we'll be having meetings with him. Thank you very much.
That's our prayer as a country. On a representative of the GC, Smart Danfodio University, Sokoto, to make his own presentation. Yes, we want to know where they are and what they are doing.
Thank you, sir. So, good morning once more. My name is Mustafa Omar Imam. Like I introduced myself earlier, I am the director at the Center for Advanced Medical Research and Training, Usman Danfodio University, Sokoto. So this should have been presented by my Vice Chancellor, but unfortunately he's somewhere else, so I've been directed to come and talk on his behalf. So although I am from Usman Danfodio University, Sokoto, but then the update I'll be sharing today is actually on behalf of a consortium comprising of 5 different institutions. And I'll walk you through the story of how the consortium came to be, what we've done so far, and then the challenges we've encountered along the way. So next slide. So the consortium is actually comprised of our university, Usman Danfodio University, Sokoto, University of Jos, a National Research Institute for Chemical Technologies area, Kaduna State, the National Veterinary Research Institute, Vom Plateau State, and the National Institute for Medical Research in Lagos. So our story started around the time when COVID-19 spread around the world. And we actually, in addition to the mortality and morbidity that was associated with the disease, we actually saw that there was disruption in so many aspects of our lives, from economy to so many things were affected. But what affected us more significantly in Nigeria and in Africa was actually the disruption in terms of supply chains. Now, other places were busy manufacturing what they needed in terms of diagnostics and therapeutics, vaccines, drugs, and everything. But we were nowhere to be found in Africa. Now, even if you had the money then, I remember when we were doing a lot of research, even then, we had the money to purchase reagents, consumables for research, but nobody was willing to sell it to us. And even if we were able to reach out to the supplier, it was difficult to actually get it delivered to us. Now, our Vice Chancellor, who was actually very interested in research, came up with this idea of domesticating some of these research efforts so that we'll also be able to begin to think of how to not only do research in the areas of diagnostics and therapeutics as related to infectious diseases, but also up to the level of manufacturing. So now the idea was to come up with a team that will be able to work on a vaccine, and then we will be able to get experts from all over the country that will contribute in every aspect that is important, right from research down to manufacturing. Next slide, please. Next, I think I've talked about this. Next. Good. So we were mandated to come up with a list of experts we needed. So this was what we came up with. We needed people with background in vaccinology, in virology, immunology. All these were actually important. And then that was how we actually reached out to collaborators that we've worked with in the past. All those members of the consortium I've highlighted were people we have worked with in the past in different capacities. Now, what we wanted to do at the time was to not only consider these expertise, but to also consider availability. Next slide, availability of resources. Now, we looked at the various institutes that we have partnered with in the past. So we considered NIMA because NIMA had a befitting clinical trial center. In addition to, next slide, in addition to various molecular biology equipment that we felt were important in developing recombinant vaccine technology. Next. Now, all these were available at NIMA. So we felt NIMA was a good pick for our consortium. Next. They had a very good tissue culture facility, which is very, very important in terms of recombinant DNA technology. Next, these are support research facilities. We also looked at NARIT, that is the National Research Institute for Chemical Technology in Zaria. Now, the reason why we considered was because they were into nanoparticles. And the reason why we were interested in nanoparticles was that we've read in the literature that there was a lot of challenges with precision delivery for recombinant DNA vaccine. So we wanted to see the possibility of not only developing a recombinant DNA vaccine, but also improving chances of delivery by formulating a nanoparticle. So that was why we brought NARIT into the picture. And then we had a befitting facility for molecular biology experiments and then recombinant equipment needed for recombinant DNA technology. Next. Next, these are all at UDUS. And then we looked at JOS because JOS had a well-built animal facility. And then there's no way we can develop a recombinant DNA vaccine without going through preclinical trial. I did mention earlier that NIMA has a befitting clinical trial facility, but we needed a facility for preclinical trials before we go on to clinical trials. And then we looked at JOS. We felt that the faculty was actually well suited for our preclinical trials. And then finally, the last member of the consortium was the Veterinary Research Institute in FORM. We selected them as the final partner because we've had experience in the past in manufacturing veterinary vaccines. Now, although they have not been involved actively in developing recombinant DNA vaccines, but then we felt being a player in vaccine manufacture with a little upgrade, they will be able to actually develop manufacture the recombinant DNA vaccines. Next. Next, these are all facilities at NIMA. Next, NVRI. Next. So what we're currently working on are two vaccine projects, the COVID-19 vaccine project and the Lassa fever vaccine, virus vaccine project. So now the Lassa, the consortium I talked about was eventually convened by the Tertiary Education Trust Fund. Now we wrote a proposal to the Tertiary Education Trust Fund, TED Fund, to receive funding to work mainly on the COVID-19 vaccine project. Now eventually they inaugurated the consortium as a national vaccine research group, and then we received an initial funding of 450 million naira. Now that may seem a lot, but then when we started, we actually realized that that was very, very meager in terms of what we wanted to achieve. I'll talk about some of the challenges later on. And then initially the idea back then was to actually work on different vaccine platforms. We wanted to see if we could be able to develop, using our various expertise, to come up with a candidate for DNA viral vector peptide subunit, as well as improving delivery using nanoparticles. Now, this other project on Lassa fever virus vaccine is solely a project in our university. Now, this received a very meager funding from TED Fund of just 40 million naira. And again, I'll talk about the challenges next. Next, so I'll just tell you a little about what we've done on the COVID vaccine project. Okay, so this was how the responsibilities were shared between the different partners of the different members of the consortium. So the idea was our university was actually supposed to work on developing a candidate for the DNA vaccine candidate, and then the viral vector candidate, while NIMA was supposed to work on the subunit vaccine candidate. NARIT was supposed to nanonize or use nanoparticles to encapsulate any of these vaccine candidates. That was, that eventually became possible to develop. And then finally, University of Jos will be involved in the preclinical trials, and then NVRI will be responsible for upscaling and mass production. Next. So what we have done, most of what I'll talk to you is what we've done at the level of our university, because that is the phase that is completely finished. The other aspects of the project are still ongoing. So what we did at Usman Danfodio University, Sokoto, was to use an approach that will increase the chances of, that will increase the efficacy of the vaccine and reduce chances of vaccine failure. Now, we've had stories in the past where vaccines that have been developed in other places, when you try them in other countries, or even in the same country, you have, as a result of mutation, you have vaccines not working. Now, part of the reason is because most times the targets that are used on the organism, on the genome of the organism, in this case, the SARS-CoV-2, is a target that is prone to mutation. So once you develop a vaccine and then there's a mutation in the target, what happens is the vaccine is no longer effective because the proteins that are used will no longer be the same as the targets against which the vaccine was developed. So our approach was to actually look at all the circulators of the SARS-CoV-2 virus at the time in Nigeria. So we looked at all the circulating strains of the SARS-CoV-2 virus, and then we looked at the spike protein. Our vaccine was designed against the spike protein, which is the commonest protein that most researchers have developed the recombinant DNA vaccines against. Now, we aligned all the amino acid sequences of the spike protein, and then we looked at the region that was conserved across all the different strains that are circulating. Now, why this is important is because we can't predict what will happen in future, whether there's going to be mutations in subsequent viruses in Nigeria, but then we looked at sequences, we looked at regions that did not have any prior mutations in the past. So what we're thinking is in future, there will be less chances of mutations in those regions. And that is the region that we have used to design our DNA vaccine candidate. Next. Okay, so at the end of the day, after we finished all the bioinformatic analysis, now we used, we did some in silico analysis, and then we came up with an ideal vaccine structure. Now, in addition to the region that we've targeted for the vaccine as our target for the vaccine design, we actually had to flank this region with two different structures that will increase efficacy of the vaccine. We used a signal peptide, which will facilitate presentation of the vaccine to the antigen presenting cells in the body, and then an adjuvant will increase immunogenicity after it has been picked up, and which will enhance the immune response. Next. Now, we did not just stop at the design itself. We wanted to predict to see now if this vaccine that we've designed, if it's eventually picked up, if it's administered into the human body, what will be the likelihood in terms of immune response? And then we looked at the T cell response, we looked at B cell response, and then we looked at cytokine production. And then we saw a very good profile in silico. So this was just using some computer models computer modeling. And then what we got was a very wonderful result. It actually indicated that the structure we have developed is actually one that will elicit a very strong immune response. Next. Okay, so this is the final vaccine construct we developed, which is in a POC57 vector. Next. Okay, so these are the different phases of the project. So the first two phases have actually been completed, which were the phases that were conducted at Usman Danfodio University, which were basically on developing a DNA, recombinant DNA vaccine. Now, we were also supposed to work on a viral vectored vaccine candidate. Unfortunately, that had to stop along the way because we did not really get good results with that. So currently, we're moving on with the recombinant DNA vaccine candidate. Now, aside from the computer, the in silico modeling that we used, we've actually done some wet lab experiments to actually validate that the construct we've developed, the construct we've developed is actually the right one. And then we've done some expression studies by using mammalian cells. We have transfected the vaccine candidate into a mammalian cells, and we were actually able to prove that it has been expressed. Now, the relevance of this is that we are hoping by this data, if the vaccine candidate is also injected into a biological system, a human or an animal, the same thing would happen by the cells that have picked up that DNA candidate to express the proteins that are associated with the target we have used for our DNA vaccine candidate. We have already passed on the DNA vaccine candidate to Zaria for nanonization. We're still waiting for update from Zaria. And then again, part of the vaccine candidate has also been transferred to University of Jos for preclinical testing. So the idea is University of Jos will test two variants of the DNA vaccine candidate, the nanonized one and the one that doesn't have any nanoparticles, so that we can compare and see if adding the nanoparticle will in fact enhance the precision delivery. And then finally, after that, okay. So when we started, we had this very big ambition that part of this funding could go towards the clinical trial, but we saw that it wasn't possible. So what we've decided was to, for this particular funding, we're not going to engage in any clinical trials. It's going to stop with the preclinical trials at University of Jos, and then the NVRI, National Veterinary Research Institute, will do some degree of upscaling before, if we're able to get additional funding, then we move into clinical trials before eventually mass production. Next. So this was when we eventually completed our own aspect of the work, the bioinformatic analysis, in silico computer modeling, as well as the expression studies. The executive secretary of TED Fund was actually quite interested that excited that he came to our university for the actual handing over of the vaccine candidate to the next members in the consortium. Next. Next. So this is a piece of news from the TED Fund website from February, showing when, celebrating when the vaccine candidate was handed from the university of our university to University of Jos. Next. Okay, so that is it regarding the COVID-19. I'll talk about the challenges in general at the end. So again, like I said, we're working solely on, our university is the only one working on the Lassa fever vaccine project. We got a funding, very little funding, 40 million naira from TED Fund. The reason why we're interested in Lassa fever vaccine is because there is no vaccine for Lassa fever anywhere in the world. And this is not surprising because this is mainly a West African thing. And Nigeria, being the largest country in Africa, possibly bears some of the biggest brunt of this particular problem. We have yearly cycles of epidemics of Lassa fever. So we're really interested to see if we could leverage on that experience from the COVID-19 project to see if we could replicate the same thing using, in the case of Lassa fever, a vaccine. Again, in this case too, we're thinking of developing, because we were quite proficient in developing vaccine. So, like I said, leveraging on our experience from the COVID-19 vaccine project, we went directly onto developing a recombinant DNA vaccine. We did not even experiment with the viral vector or the subunit vaccine. We went directly onto the DNA vaccine because we felt that at the time we were actually very proficient in the techniques involved in the vaccine, a recombinant DNA vaccine. Again, we use the same approach as we did in the case of the circulating strains of COVID-19, circulating strains of SARS-CoV-2, the causative agent for COVID-19. Now, in this case, we looked at the literature and saw that there were so many lineages of the Lassa fever virus. There's about 6 of them circulating across West Africa and Nigeria. There's 1, 2, and 3. But then we did not only want to focus on 1, 2, and 3 in Nigeria. We looked at all of them. We aligned the sequences of all of them, and then we looked at conserved regions across all these lineages, such that the idea is, there may maybe one of the lineages that is not very common in Nigeria, but somehow somebody will still transport it to Nigeria. And then if you have a vaccine that is just effective against 1, 2, 3, not against 4 or 5, because they're mainly found in Mali or Sierra Leone or Liberia, then if somebody imports that particular lineage, it will be a problem. It will be a problem for us. So we designed our DNA vaccine candidate taking that consideration, taking into consideration the fact that it should be effective against all the lineages of the Lassa fever virus. Next. Okay, so this is these are the different phases of that particular project. It started with, just like the COVID-19 project, started with the bioinformatic analysis and then the construction of the DNA vaccine construct. We did some wet lab experiments to see if the construct would be taken up by mammalian cells and the products are going to be expressed. And then eventually we will do immunogenicity and preclinical trials in animals. Next. Yeah, this is the final vaccine construct, again in POC57 vector. Next. Okay, so just like we did with the COVID-19 vaccine construct, the candidate vaccine, we did the same too, just so that before we begin the preclinical trials, we wanted to be sure that using computer modeling, we will be able, our vaccine candidate will be able to elicit a very strong immune response. And then we looked at T cell response, B cell response, and cytokine production, and we saw very wonderful results to show that our vaccine candidate is likely going to, in a complete biological system, will likely elicit a very strong immune response. Yeah, next. Okay, so this is what we've actually achieved so far. All the phases of the project have been, we've successfully completed them. What we're waiting for is the preclinical trial. And the reason why we've not been able to complete some aspects of the preclinical trial is because of some of the challenges associated with handling the Lassa fever virus, which I'll talk about subsequently. Next. Okay, so we've not only been doing the walk. We've not just been doing walk. We've actually been actively involved in also spreading the news of what we've been doing. So we've actively been, the members in our consortium have actually been very active in publication in the aspect of recombinant DNA vaccine production. Now, the reason is because we want to actually, bring the news out to the world that we're actually doing this in Nigeria and it's possible to domesticate some of this expertise in the country. Next. Okay, so these are some of the challenges we have faced for both projects. The biggest challenge we have had is that of funding. Right? Like I said earlier, when we received the 450 million naira from TED Fund, we were very excited, thinking this will go a long way. But then by the time we began to purchase the consumables, the equipment, and the resource, we felt that wouldn't go anywhere. So we had to actually bring down our expectations. Another problem that we have had is the Naira dollar. This has affected most of our research projects, not just the COVID vaccine project. What we've noticed is in Nigeria, the time you apply for funding to the time the funding is actually released, it takes a very long time. Very long time. So the problem is the dollar rate that you use in your application would have changed. And then because we don't manufacture any of the consumables, the reagents that we use in the country, we have to purchase them from outside. So the problem is if you, there was a particular grant, I think it was the last one we got, by the time we received it, more than 25%, the value of more than 25% was gone because of the increase. We're meant to understand, at least we've discussed this at various levels in the university, is that research equipment, consumables, and reagents are supposed to receive some degree of waivers or some special consideration. The problem is we purchase reagents. A single tube of a cell line, for example, sometimes we buy at 3 million naira. And when we tell people we buy cell line, which is a small tube, 3 million, people can't believe this. They think that we're just inflating the value. But we have purchased cell lines sometimes up to 5 million naira. But the problem is when those cell lines arrive in Nigeria, in Lagos, we are in Sokoto. We can't begin to travel to Sokoto just because we want to receive a cell line to Lagos, sorry. Now, the people especially NAFDAC, they receive our items and they just keep them in their office. And then when we try to inquire, they tell us to come to their office. We can't leave Sokoto to go to their office. And then most times by the time those items are cleared, we've finished all the paperwork and everything, we receive the items and the cold chain is not maintained. So we have lost a lot of money as a result of this. Yeah, and well, we've had a better experience with customs, but they've also been doing this to us. They receive items, and then we really can't understand how this is happening, because we've inquired and they told us that because we mainly ship via DHL. We have an institutional account with DHL. So when items arrive in Lagos, we receive an email or phone call that customs has confiscated, held our items. As soon as we finish with customs, they tell us that they've transferred it to NAFDAC. So we really can't understand why this is so. I mean, most of these things could have actually been prevented by informing us upfront that this is what we're supposed to do. Sometimes we even try to do that, but we don't even get any response. Yes, we've done that because we have an institution. We go through the DHL manager and so to let them know that this should be allowed to come in. Yeah, so but we've had serious problems. We have lost a lot of money, research funding, just because of this issue of importation. Yes, so again, another problem is problem with power supply. We have, like I mentioned, the fact that we buy reagents sometimes that are very, very expensive. A single tube can cost 5-6 million naira. We bring them to the lab, and sometimes we don't have the electricity to actually power our refrigerators. So what we've had to do was make a case to TED Fund, because the original funding we received from TED Fund did not include generating alternative electricity. So we made a case to TED Fund that we have to use part of this funding, because everything will go to naught if we don't use part of the funding to generate electricity. So right now, this has partly been solved because we have dedicated backup inverters, solar panels, and yeah, this is actually solved. Okay, another problem is that of lack of a befitting animal house. Although University of Jos veterinary faculty has a very good one, but it is still not ideal for testing of vaccines, because when you want to use an animal facility for testing vaccines, it has to be well controlled. Even the air that goes in has to be filtered, and it needs to be well kept. And that is the case in University of Jos. So yeah, it's been a problem for us, even though that is what we've been using. NIMA has a clinical research center, but then even that actually there are just very few beds for clinical research. So it's actually good to have some of these clinical research centers spread all over the country. Can I have my slides? Okay. Okay. So anyway, these are some of the challenges and some of the way forward for research, at least I'm talking from a research point of view in the country, not just for us, but for any research team involved, interested in research, is for there to be additional funding for research, not just for vaccine, for every other aspect that is relevant to national development. Now, when I did my PhD in I saw a scenario whereby not only the government was providing funding for to universities, but even the government was actually, as CSR, corporate social responsibility, the government was making big companies to actually provide research to universities. The project I worked on was $1 million project, and it was sponsored a private company in Malaysia. And this happens, in so many other. So I see no reason why in addition to government, other private entities in Nigeria cannot be made to provide funding for research. Again, this is not saying that government shouldn't also do better, because so many other ministries and agencies in the country could actually provide funding for research. Again, the other way forward we see, especially with regards importation, is for there to be, I don't know, maybe if there's a way NAFTA, customs, and research institutes in the country can come together to develop like a framework that will make it easier for us to bring in items. Because as it is currently, we bring in reagents and we have to pay exorbitantly for customs duty, we have to pay for NAFTA clearance and so many other things. And sometimes by the time they get to our lab, I mean, they're gone exactly. There were times when we had to sponsor somebody to travel outside the country and hand carry our reagents back to the country because it was safer for us. Because whatever problems there were at the airport, that person will resolve it with them and then bring it to the lab. But most times, if it comes in through Lagos, we've had this problem several times. Especially if it comes in around Thursday or Friday, that's it. We are sure we're going to lose those items because they don't properly store them in cold storage, and most times it has to be Tuesday or Wednesday before we even are able to reach out to the right person to be able to clear it. And we have spoken to one of our partners because we thought maybe it was because we were in Lagos. That was why, in Soko. That was why we were having this problem. But we spoke to NIMA. The same problems we're having is the same problem they're having. We've actually had stories that our partners in NIMA, that's in Yaba, Lagos, there was a time they brought in very expensive reagents that needed to be stored at -80 degrees. They went to discuss with I think NAFTA on a Friday, and they were told that the person in charge is not present, and so they can't be given those items. So it was after pleading and high-level discussions, they were allowed to take those items, go and refrigerate them on a Friday, but they had to write an undertaking that Monday morning they will bring them back to the NAFTA, and then they had to bring them back, and then back and forth, they were able to. Now, if this had happened to us, there was no way we could have to Lagos to take those items, put in cold storage, and bring them back on Monday. Yeah, so these are, okay, again, so the issue of BSL-3 and BSL, we are working with very deadly viruses, especially Lassa fever. Now, most times we have to use activated virus or a pseudovirus. Now, we don't expect a pseudovirus or an activated virus to produce the same type of immune response as a live virus. So this is one of the challenges. We've had good results so far in our experiment, but we feel if there is a BSL-3 and BSL-4 in particular for the Lassa fever, this will probably help us to generate better and more reliable results. Yes. Okay, next. Okay, so well, I think the take-home message here is that there's actually local capacity for vaccine research and development, like I have highlighted in our choice of partners for our consortium. We actually had to look at what expertise each member has and what each member is. I can tell you, over the years that we have collaborated, we think all these partners have, although we have not had response as we had wanted from some of the partners, but we think they have the right expertise, and with the right degree of funding and the right motivation, will actually be able to achieve a lot in the country. At least we've shown this with the COVID-19 and the Lassa fever DNA vaccine candidates. Again, there has to be massive amounts of fund for vaccine research and development. Without that, whatever expertise we have locally will actually mean anything. And then on behalf of the members of our consortium, thank you for listening.
Thank you very much. I decided to
stand up. I may ask a question or make contribution. I don't know what it will be. Let me just put my thoughts together. But when I go to the slide on the membership, your partners team, that was when I knew I have to. Even though you started with supply chain, business of everything, there's nobody in that team that will support you on supply chain. And there's nobody on regulation that will support you on that team. So I wasn't surprised you had that problem with now, because you didn't prepare for it. That DHL, whose primary goal is to move commodity from one place to the other. And you concluded in your one but last slide that you are handling dangerous entities. And finally, when you produce that vaccine, if it gets to the market, you are out of the system of what is happening to it. Is the supply chain specialist that gives you visibility into the pipeline. Because the life cycle of that product is even more, higher percentage is in the market, in the supply chain feed. So I see that as where we have challenges. If you had included in your protocol the rule on considered regulation, you would have been provided all the requirements because you would have listed all the commodities you will need. So even if your commodity arrive on a Saturday night when people are in the clubhouse, it will still be taken care of. See, this I'm telling you, my organization when I joined in 2016, enough that used to seize our commodity, everybody was blaming. But I can tell you that incidence has reduced drastically. Because there are procedures you go when you are bringing such commodities in. You should have pre-informed that there is a porter, even the trade porter. Your commodity should have handled. When you have third parties, because they want to make more profit, they ignore that part. You said? They ignore that part of it. So I see that, and I see that there's a gap which will not be considered. I'll be presenting to you the whole thing. Regulation takes far very high importance, even your ethical approval. So I think for future, we need to, because each establishment has mandate. And you can't, because you are bringing 10 million naira out of the 450 million to bring in one tube, say I should not follow my own ethics. It is better, we both achieve better if we understand each other's role. If I can contribute to when you were preparing, so that, in fact, I know that by June next year, you are bringing this. So there's nothing that will stop it. They are not saying don't bring. And there are also provisions. I decided to say this before NAFDAC. So I told them, see if NAFDAC is trying to defend themselves. I work with them a lot, I support them, and I've learned the rope. Now my brother there, MPACD, if you ask them, MPACD and NAFDAC, they are twin sisters, they fight. They used to fight. Yeah, they used to fight. But everybody has to understand, there are provisions for what you are doing. But you cannot come to my house after you have defecated and now saying you're knocking. No, it's not done. If you apply for waiver, they will give you the waiver. In fact, there are some products that they need to inspect, particularly if it's coming from China and India. before they leave. But you bring them and it is our doorstep. That's why I say NAFDAC is holding you. They will hold you. And there's nothing you can do about that. The best thing is when we are planning, they may not even be statutory as partnership if you don't want to recognize them, but involve them in planning. Let through them understand your regulatory requirements for all the things you'll be using. Meanwhile, you have a beautiful presentation. I'm proud to be a Nigerian for what you guys have done. Thank you.
Because I know NAFDAC was hit below the belt, but we want to protect them. Above the belt, above the belt. Thank you so much, Professor Musk, for that beautiful presentation. In fact, the moment you started presenting, I felt, are we in this country? Sokoto is so far away. Sometimes I from Niger, the part of Niger itself. But to see that you have a team starting an important project in Sokoto, it makes me happy. I've also heard of some of these efforts from other universities. And I ask, what are the other universities doing? Really? If Sokoto can put up that kind of effort, I think it is something that we're proud of and we should clap for them. So sir, my brother has already spoken about team because that was the first thing I noticed here, team. Your team has to be reviewed. When you put only eggheads there, you think that everything will go well. Work like that. In fact, if you can bring a senator, a politician, and put there, it will even help you. They may not understand the scientific basis of what you are doing, the RNA, DNA, they don't know, they don't care. But there are contributions they will make that will make your job much easier. And you also need to bring in financial experts to help you in your budgeting proposals. You produce a budget of 1 billion and they're giving you 40 million, for God's sake. Do you need to even take it? And you see, the process of budgeting is very, very intensive. You have to capture virtually all the components of the cost. And you should be able to defend exactly the components. And you tell people, use Professor Nasidi, he is good at that. Get him involved. I can assure you, if Professor Nasidi were to present anything for you before anybody, unless that person is the devil himself, he will listen to him. There are people like that. Look out for them and get them involved. Like I said earlier, it's not about just one team doing something. You must reach out to others. And I think I mentioned that. It's very important. Nobody is insignificant. Nobody. And NAFDAC is one of the pride agencies of Nigeria. And we respect NAFDAC a lot. They may have their own challenges here and there, but believe me, they have to do what? Excuse me. They have to do what nobody kept from here. They have to do what they are doing in order to protect us, to safeguard our health. And I really thank NAFDAC for standing firm. But NAFDAC, because we are defending here, doesn't mean that. Please, for the sake of our country, there are certain things that you have to just ignore in order to make progress. There are certain things that they have to probably take easy and make it easier for people like you who are in serious research. And I think it is very important for you to also understand that research work of this nature, even if you don't have all the components, you must continue to push. You must continue to push. I wanted to ask questions about the scientific aspects, but I know it will not be appropriate at the moment because quite a number of processes are involved in vaccine production. In those days when we were in school, were told that we knew that before one candidate product can come out, it will take between 15 to 25 years. Now we're trying to bring out even vaccines in how many months now? 4 months. Let me be honest with you. For some of us who are old school, we cannot just come to accept this reality. You must make efforts to try and get people to appreciate the science that goes into this, and to also let the people know that safety is taken into consideration in all these aspects, so that when the candidate vaccine comes out, people will not be resisting. My wife was in Sokoto for this their immunization thing. And believe me, there are pockets of communities in Sokoto that are still refusing to take. And Sokoto still presents the highest in the whole world, the highest incidence of preventable problems not just in Nigeria. And people are refusing to accept it. And I thought I should mention it because being an elite from that area, you must help the government. If not so, by the time you finish all these science and laboratory things, the candidate vaccine comes and people will refuse. Of what use would that be? We will just be spending money, spending efforts, your brains will be up. At the end of it, you bring out a vaccine, nobody is using it. And to start from Sokoto, and that is not fair enough for all the efforts you are putting in. So as you do all these things, you have to gather all the various, have to understand the politics behind all this issue of vaccine production, drug production, whatever it is. I don't want to take, there are so many we should allow. Thank you very much for that beautiful presentation.
Thank you, sir. Thank you, Professor Mustafa, for that presentation. And I want to say thank you to our father in the house and Mr. Hamzat. He has already defended NAFDAC well, so I won't say much. But again, like he said, and I think that is part of why we are here, collaboration. Collaboration. We need to stop thinking and working in silos. We are one government and one people. So we need to stop working in silos. The incident you mentioned that happened with the colleagues in NIMA, I don't know how long ago that was, but I'm not sure they can say they still have that same experience now, because we've had to work. He's from NIMA, he's just, he's doing his sabbatical in USP. So we've had to work together closely on this kind of issues. So like he said, it's for you to let us know in advance. You need to let us know in advance. There is a government portal for trade, trade.gov.ng. That's where we key into it, custom keys into it, CBN keys into it, for all, And then when you already let us know, we give waivers. It's government, it's research. There was one Dr. Amo in Naima. He told me he paid maybe 1,200 naira or something, just some fee that is to clear the goods, and I think it was a customs thing. He didn't have any problems with NAFTA. 1 naira. You see, so it's for you to let us know ahead. We've had several cases like that, but that's in the past. The portal is online, 247. So it's for you to make your request and all of that. And then again, like I said, collaboration. Please, and this applies to all other research institutes, please let NAFDAC know what you're doing. Let us know what you'll be bringing in. And at times, some of these things go beyond our own mandate, especially when you're bringing in these cell lines and all of that, because of bioterrorism. Because of, you also, there are times you need to also liaise with the office of the NSA and all of those kind of things. So when you do all of that ahead of time, by the time your items are arriving, you shouldn't have any hitches. So please, that's just the main thing, collaboration. Let us know ahead of time, and then you would receive all the cooperation. Thank you.
Yeah, good morning. My name is Yoms Ishaku. So my others have said it all. And just to add my voice that last week, the Director of Food Drugs Services mentioned that all IDEC are expected to be submitted in our office before the end of 31st January. Madam Selin, you remember what the director said last week? All import exemption certificates to be processed by NAFDAC are expected to pass through our office latest 31st January. For the group, that department will collate for the whole country and get approval on behalf of all agencies and organizations in the NAFDAC office. And with what WHO has said, we manage the immunization supply chain commodities for the country. We have cold chain equipment scattered all over the country, from the national zonal store states. In Sokoto, we have many of them in even LGs, but then they are basically for immunization, for our own vaccines, unless we are being directed by the federal government and it has to pass through some processes before we can now. But however, like your reagents that can be stored around minus 80 degrees, there are so many equipments that we store these COVID vaccines that are around that minus 80 to from minus 25 to minus 80 degree. And since the COVID vaccines are fast growing now, the only one we have around now is just the J&J, and the equipment are not and they are not occupied. Basically, we use them for production of ice packs for transportation of vaccines. Thank you.
Good afternoon, good morning, good afternoon, everyone. All of the above. Sorry, I came a bit late. I'm Pharmacist Hasia Dara, a Deputy Director at the Pharmaceutical Services Division, and I'm one of the focal officers as regards IDEC in the Department of Food and Drug Services. So just as Pharmacist Omota mentioned earlier on, I think this, where we have issues is, people applying for IDEC or for the import permits from NAFDAC. Usually the major problem we've noticed is that people don't know where to go, they don't know who to contact, they don't know the process, they don't know anything about it. So most of the times these things come into the country before they even start moving around. For us at the Federal Ministry of Health, we process the support letter which goes to Finance, Ministry of Finance. So Finance issues the IDEC as regards that's the import duty exemption certificate, and then we also write to NAFDAC for the import permit. So what we usually advise our partners is that they should apply on time, because usually sometimes the minister has to endorse these letters, and due to official engagement, sometimes he may not be on seat and we may have little delays. But when we have, so collaboration is really the key. Knowing what to do, knowing who to meet, everything at the right time. But it's really important to start the process on time. And just this year, we started having a waiver stakeholders meeting where we bring all the players. NAFDAC is there, we're there, Finance is there, Customs, they're there. So we bring in everybody that's involved in the whole waiver process, share ideas, challenges, and these big players there in the room. So they answer these questions. And I think like Finance, they've automated their process. So it's much easier now within a shorter period of time to get the IDEC. And over time, over the years, we've noticed that some of these hitches we don't, they're like becoming a thing of the past. So I think just knowing the right thing to do, and then it's really key to apply on time. Thank you very much. So I think in the future, you may not have these issues.
One of the it's a peculiar situation because he is from the university, which is clearly of education. I don't know if they are involved in all these are likely to
They're involved. So we're going to have another meeting next year. So we'll invite, we'll definitely, I'll get your number, and so that's the
Okay. Yeah, just. Thank you, Professor Mustafa. I mean, one is actually excited from the industrial point of view about this effort that you are putting, that the consultant is putting on about local vaccine production. One of the things that struck my mind in the presentation was the fact that you referred to the preclinical laboratory that you have even identified that it's not up to standard that you desire. I want to suggest, without making too much about the fact that you need to collaborate with NAFDAC. NAFDAC is the regulatory agencies that set up standard for production activities, either in form of these preclinical trials or even manufacturing. They will be able to guide appropriately in terms of what we call cleaning facilities, the kind of airflow that you have, velocity, and various other things that you need to use in making the construction of that thing, so that your effort from the beginning will not be trashed at the end. So that's just my contribution. Thank you.
Thank you so much. Mine is Nemeka Ndondo. I want to actually commend my brother, Dr. Professor Mustafa. What he's talking about is very exciting, very nice, but good food, good movie, I'm really killing. You know, when you see somebody was talking about Sokoto, his farm. Now, when you see products coming up like that, it takes serious leadership. When you have good, strong leadership, you make investment. If you don't make investment in science, you're not going to get anything from it. Massive investment was made in that center. That is the director. Massive equipment were put in place there for them to do because they were forward thinking. So we are so proud of what they are doing. Actually, I started this, I was the director of the center and he took over from me. So he's doing amazing things and much more great things will come from there. But I have some things to, like a comment. I've been doing great things, and unfortunately, I've not, because of I was so involved with NCDC, I've not really got to know much of what they are doing, but right now we're very much involved. One is validation of the in silico targets. So it's very, very critical. I don't know whether you have actually done wet lab validation of those targets. It's important for us to know. And in saying that, there is great hope for Nigeria because we have also, working collectively, we have made plan to have these things. This is another rocket science. It's seriousness, investment. You can't reap without making robust investment. He's talking about to do this kind of work, you're talking about $400,000, 400 million. It's nonsense. You know, we have to be serious. When we are serious, we spend money on science. And when you spend money on science, you reap from it. You know, somehow we made investments right now. We've dropped a lot of equipment that will do most of the things I'm talking about to validate those targets from a logical point of view, from every chain all the scientific chain he needs to do, you know, to do that. You know, so again, working on BSL-3, for you to be handling things like Lassa, it's not something you'll be dabbling with at biosafety level 2. It's very risky. In places where they do those things, it's just to have a good biosafety level 3 lab constructed. Fortunately, there's one that we are hoping that will come up soon by Japanese government, JICA, that didn't work for NCDC currently. We hope that such things will see the light of the day, and they are seriously doing the construction. Maybe hopefully this January they will do that. And why doing it? In the animal facility we are talking about, the animal facility has to, for you to work in Lassa, has to be in like a biosafety level containment. You don't just put cage inside and you're putting Lassa there. Like in Japan, that's what they do. You have all the biosafety level 3 containment will have animal facility in it where you do those type of things. You don't try just doing it. Although you can practice biosafety level 3 containment if you don't have the full laboratory. So it's very, very important to really handle this virus at an appropriate level. But for you to do actual Lassa work, we need to have a biosafety level 4, but let's have level 3 first, and get it properly working. The power solution. Power is a lot of challenge. I know in that facility they have plan, but to run it, maintain the fuel of that place, because the vision of the place is wonderful. Dedicated over 250 kVA generator for that center alone, but that was not enough for what we need. So something like lithium power backup, plenty of it. In the project I was talking about, we ordered 40 units of that. One of them can carry if you can have like 2 or 3 of those units, you won't be talking about power if you link it to solar. Then another thing is need for sequencing of Lassa. You've talked about lineage 1, 2, 3 around Nigeria, 4 and 5 in other parts of African country. So I've noticed that really sequencing of Lassa is really a project that we need to do to actually understand what is circulating. Which ones are the one that drives severity? Which one is the one that drives survivability? These are the things we are not under. So that we have to make robust investment in genomics facilities to be able to do those things, so that we'll be able to know our own sequence, be able to adapt it, like what happened in South Africa. When they are building the license they got from biotech, they were able to use the sequences, not the original sequences, but the other people developed. So we need to have that kind of robust capacity as we plan. Thank you so much.
It's my name. Thank you, Dr. Mustafa. I know of your setup, Professor Mustafa. You must have heard of me too, from James and so on. So I'm doing my sabbatics with USP. So this is the question I've been asking the NIMA team, and I have a reason for it. Let me start with your basin construct. Why are you using interleukin 12 to enhance immunogenicity? Why not interleukin 2? Because of the helper function of that interleukin to make antibodies where we are hanging our protection. So I've suggested to them that they can create a hybrid in that optimization, do IL-12, do IL-2, and repeat the immunogenicity prediction, the cytokine profile, the cell-mediated immunity, and humora. And let's see whether they will be different. Now, the second one is the animal facility. I must tell you, the NIMA team tried. They got it once in terms of raising antibodies against the spike protein construct that they made, and they could not repeat it. And there is answer for it. You see all those things that we read in the literature in terms of recommended animals to use to test. Yes, I want to tell you that they are also genetically engineered. For instance, the rabbit model for boosting IgG against SARS-CoV-2, when you make your construct, they've engineered them so that they could have abundance of ACE2 receptor. So you don't have to only look within, but work with, collaborate with companies abroad that make that, like Jackson Laboratory, so that when you put them there and you use the African initiative now to midwife that, we can get those animals at lower cost and do it. Relying on our own breed, you won't get results. So you won't get results. The one for NAFDAC, I think, has been addressed. So NIMA are the same thing. when they were trying to make inactivated vaccine, there is one chemical that they could not get. They said NAFDAC seized it one way or the other, but the trade portal worked. But what I will also want to say is that both the Department of Food and Drug Services and NAFDAC, they need to come out and give us the framework and necessary forms to use. Even if we are using the trade portal, if I open it, is there a form? How do I sign it? How do I get it approved? If we walk through that ICT, we get it easier. So how do I register? You know, we have to create an account, the PI, you get it, they give you a password, and so we need that system to streamline the process. Above all, thank you for that wonderful presentation. Thank you.
One small question, sir. You were mentioning team. After you mentioned it, I wanted to find out, are you collaborating with other teams outside of Nigeria or just local teams? Because sometimes we have to steal this technology from outside. If you are collaborating with, you should be able, he mentioned just one now, and like it occurred to me that you need to probably look at outside and find out who can be of assistance, who will be ready to support us, even if we have to pay them, you know, to do mercenary work for us. Please think about that. Yes, sir.
Yes, just to add to Prof. Toward, even the forms you are requesting for, PID does training periodically. In fact, I think last week I got another invitation for it. So if we talk to NAFDAC, they will tell you when that training is coming up. They will take you through the whole portal, what you need to do there, how to monitor it. In fact, for us now, we are priority claims. If I have issue, I go to the WhatsApp group, I drop question there, and get attended within the next few hours. I get attended to. So please, I think the word she said, collaboration, collaboration, collaboration. Incidentally, my own presentation is a pillar there. And I think that's a big challenge in all aspects of what we have been doing. From sticker manufacturing, vaccine manufacturing, everybody is running their race. It's not a 4x4 race we're doing, and that's why we're not getting the gold medal.
Again. No, sorry, just final one. I wouldn't talk again. The issue of waivers, I read recently, even the National Assembly is raising concern over waivers. Now, personally, what do you mean by waivers? Since 1994, I've been fighting on this issue of waivers. I do not support waivers. It's inimical to anything we're doing. Waivers, because of waivers, we've lost billions of naira worth of goods in the past. And so the processes are there. Finance will give you, NAFDAC will give this. I'm telling you, there's nothing like they will give you. Every time we went for waivers, we had to pay. Meanwhile, the items are there in the post and they're deteriorating. And I'm happy that the National Assembly is looking at that. They're paying waivers to who? It's government to government. If you are going to pay for anything, make provisions for it. As soon as the items arrive, you go and pay and collect that thing. The money is going back to government. It's government money is going back to government. It's just because we are too corrupt in this country. That is why we're having problems. How it is government money? You are taking government money to go and give government again. Is there anything wrong with that? But when you make provisions for waivers, you introduce the element of corruption. The worst type of corruption that I've seen is about these waivers. It is not right. It favors some few people, and it is a huge disadvantage to so many other people. And it derails the programs, such as the one that Professor Mustafa and his team had. I am opposed to waivers, and I will always oppose. Thank God I did not become a senator. because believe me, it was one of the things I was going to fight against. If you are going to give government money to government agency, what is wrong with that? Just make provisions. As soon as the items come, you pay the necessary fees, take your items. I hope people would look at this, because for many years we have been talking about waivers. It's not helping, and people are suffering. Government resources are just going down the drain. Some people are pocketing money because they want to take money from you before they do anything. These are things we have experienced, man. And for many years, we tried to see how we could go around it, but it didn't work. I just pray for. Thank you.
Can I respond to some of the comments? So thank you very much for all those comments and questions. First of all, I would like to respond to Mr. Hamzat, right? Okay, yes. So he did talk about some of, yes. Okay, okay, fine. So in response to your comments regarding the fact that you don't quite trust the process of the current recombinant vaccine development timelines, right? So I want to believe part of the reasons why we're now able to develop vaccine candidates within a shorter time frame as compared to previously is because of the availability of modern techniques like bioinformatics in silico. But then in our own case, we're not trying to rush it as we have seen in other places where they were able to come up with vaccine candidates from the lab to the bedside within 1. And possibly that is why we're having problems now. In the US, for example, there are so many reports of vaccine-associated side effects, even deaths. A lot of people are having cardiac complications, neuro complications, and even death. That is possibly because they did not take their time to look at the side effect profiles. Now they quickly injected it in people, and now people are having problems. So in our own case, we're going to take it very, very slowly. And even our approach is quite different from what they've done. I can understand the fact that at the time they were in a rush because they needed, COVID-19 was ravaging through various countries in the West, and a lot of people were dying. So they needed something to quickly administer to people. But in this case, we're not in a rush. So we're going to take, even at the stage of animal trials, we're going to take it very, very slowly. We have to go through the immunogenicity testing, vaccine efficacy. We're not in a hurry. And just like it was mentioned regarding the animal strains, yes, part of the challenges we're having now is the issue of the animal strain. We do appreciate the fact that using our local strains may not quite give us what we want. So we've been in talk with Jack's lab in the US, but the cost of those transgenic animals is exorbitant. Exactly. And then another thing we actually looked at is the fact that if we're even able to pay what they're asking for to purchase those animals, if the moment they arrive in Nigeria, they're going to die because we don't have the befitting facility to keep them. They need to, the facility needs to be contained. The air needs to be well filtered. It needs to be very, very, well regulated. So we don't have it. So at the moment, yes, we're using our own local strains, but eventually, like I said, we're not in a hurry to rush into clinical human trials. We will eventually have to use those transgenic mice, animals. They're already talking with a lab, again, regarding the issue of collaboration with international partners. When the idea started of this consortium, we specifically did not want to bring in a foreign partner. And I was one of those that said, no, we shouldn't bring a foreign partner because now from our experience, most times when we collaborate with people from outside Nigeria and we don't bring anything to the table, they tend to look down at us. Even if they don't show it, overtly, they will definitely look down on you because you're coming with open arms trying to take from them, but you don't show anything in return. So what we are thinking for this consortium is before we will eventually invite collaborators internationally, but we want to be able to have some robust data to show them that yes, we have this expertise and we can do it without your help. All we need is some additional support from your experience to be able to upgrade this. For example, in the case of the animal test, we can't do it. So we need people from outside. University of Jos is already talking with them. They have collaborators from Germany. In talking with them, they have a BSN level 3. So part of the experimentation will go to, will go to Germany. Yes. So, okay, yes, sir. Okay, yes. Anyway, I was just going to say thank you for your comments. And when we started this project, we were quite excited. Some of those members that we should have included, we did not, because we did not know they were important. But now we know better. So moving forward, we're definitely going to consider additional partners if we're going to, such projects. On the issue of wet lab validation of the in silico targets, yes, we've done that. We've actually had to look at, we did not just rely on the data from in silico modeling, because using that may be misleading. So whenever we had in silico data, we actually had to validate. So for example, after we had the construct, we actually had to use, we had to use PCR to confirm that the construct actually contains our target, the target when we're trying to. And then even after that, we had to use restriction enzymes to cut, to see if we're able to get a pattern that is representing the target that we've included in our construct. Yes, we did. Exactly, we did. We did not do it in our facility. We outsourced it. We sent it out for sequencing, and that is also for the validation, right? So we did PCR, we used REs to confirm based on the band sizes that were, the different fragments, and we also sent for sequencing. So yes, we did validation of the targets. Again, in terms of lithium batteries that you mentioned, yes, we're now actively, installing lithium batteries. Yeah. So in addition to the backup that we have, the generator backup, we have a lot of lithium batteries all over the center now. So we could actually now go without electricity. Our storage facilities, we can run our experiments without any electricity, without putting on the generator. So regarding the use of IL-12 and IL-2, it wasn't my area to decide which one to use, but what I can say is we actually used a lot of, using the model, we used so many different adjuvants. And then the choice of IL-12 was based on the profile that we got after T-cell response, B-cell response, and cytokine production. So we did not just blindly use it, we used so many, and based on the in silico modeling, we saw that was the best, so that was why we used it. Of course, moving forward, we're open to improvements. Moving forward, if at the level of the animal study we see challenges, we can easily just come back and try to improve the construct by possibly changing the sequence that we've tactically and the adjuvant. Anyway, that's all I have. Yes, it's okay. All other comments and questions will be cheated. Thank you.
Okay. Thank you so much. Who says we have high level people here, high level engagement, and we are meeting our targets? That's good.
Thank you. Sir, it's a response to the waiver that you mentioned. The National Assembly, they're saying so much as to why the country is giving waivers, the country is losing much in revenue. We see that, it's not just government that applies for waivers. Other organizations apply for waivers also. With respect what's captured in the fiscal policy measures of Federal Ministry of Finance. And I can give you an example with Medicine Sun from Tech, that's Doctors Without Borders. They started, I think, 2014 their presence in the country. It started with like 6 sites scattered across, upgraded to 8. I think currently 10 projects across Nigeria. And they're doing quite, and everywhere for labor, the IDEC and the imports coming from finance. And they're doing so much in the health sector, helping our people. I know they're doing what government should do. Yes. You know? So I think unless government sits up and puts the interest of Nigerians at heart to do, because personally, I don't think that as giants of Africa, we shouldn't be relying on support from partners. I think they're big enough to take care of Africa. So for me, it doesn't even make sense that let's so much money. This is the National Assembly that's spending money on things that really improve the life of Nigerians. So if other people are doing the health sector different. So I think as regards the waiver, I know the country is losing much in revenue because the waiver is a percentage of the total commodity. But when we look at the other aspects of it, Nigerians are we need to learn from other countries. One part. And you know, just as saying that